Welcome back. We just finished talking about mitosis. We'll do a quick review just to keep it straight in your head before we move on to meiosis, which is a different kind of cell division. So get your coffee, get your pens and pencils and your notebook ready, and let's get right back into that chapter. So it always makes me start at the beginning. Let me get to where we were. All right, we had finished talking about mitosis. Here's just a little overview. Again, we looked at so many different pictures of how it all works, right? So prophase is that nucleus dissolving, the chromosomes condensing. We can see them now. And you don't need to worry about like proto-metaphase versus metaphase, okay? Like don't worry about that. So some of these in-between phases, we can just call this metaphase, okay? We can see our spindle fibers. The chromosomes are all lined up in the middle, and each chromatid is attached to a spindle fiber. So if each of these is a sister chromatid, each one is connected to a spindle fiber. They're going to get pulled apart to different sides. That happens during anaphase. And then telophase, we've got our new nuclear membrane forming. The chromosomes are going to start to uncoil. They're going to be less visible now. And cytokinesis is just dividing everything up in the cell, right? So outside of each of these new nuclei that have formed, we've got some endoplasmic reticulum, we've got some other organelles, okay? Divvying all that up, that's cytokinesis. Okay, and you can see that it's different in plants and animals because animals do not have a cell wall and plants do. And we're actually going to look at this in a little more detail in lab. But for animals, right, you can just pinch it right off. It's like a balloon, like that you can just squeeze and pinch, and you've got the two halves, right? For plants, because they have that cell wall, if you have a room, right, and you want to divide it, you can't just pinch your room, right? You'd end up crushing your wall. You need to build a new wall if you want to divide it. So that's what happens here. There's this cell plate that's put in. And once that reaches both sides, like it starts in the middle here, it moves out to the right and to the left. Once that's formed, then they can divide, and put a new cell membrane in. So for animals, cytokinesis happens from the outside in. Right, it starts out and it pinches towards the inside. And for plants, it starts at the inside and moves out, right? You start with your little wall, this plate in the middle, and you move to the outside walls. Okay, and when we finish mitosis, at the end of this, each cell in the beginning, that one cell that we started with had 23 chromosomes. Each one had 2 copies, so we had 46 total. Of those 23, of those 46, 23 are from your mom, 23 are from your dad. Okay, that's what we started with. We divided each of them, or replicated each of them, right? So we ended up for a little while, if this was one chromosome, we now have 2 copies of it, right? And this is chromosome 1, this is chromosome 1. We now actually have 4 copies of chromosome 1 instead of 2, right? So for a brief moment, we had 92 chromosomes. And then we divided each of those, and we're back down to 46. So we started with 46 chromosomes. We end with 46 chromosomes, 2 copies of each. You still have 1 from your mom and 1 from your dad. You copied your mom's and divided it. 1 went into each cell. You copied your dad's, divided it. 1 went into each cell. Okay? All these cells that go through mitosis are diploid. This means they have 2 copies of every chromosome. They have doubles, right? You have 2 copies of chromosome 1, you have 2 copies of chromosome 2, and in a cell that's not ready to go through mitosis, each one is just one long strand, right? You've got 2 copies of 1, 2 copies of 2, all the way through, 23, okay? These are diploid cells. All of the cells in your body, except for eggs and sperm, are diploid. They all have 2 copies of every chromosome. So that is what's known as a somatic cell. It's a body cell. It's not a reproductive cell. It's just one of the other cells in your body. So the vast majority of your cells are diploid. Now, we talked a tiny bit in the last lecture in the beginning how it goes through these checkpoints to make sure when cells are dividing that that the DNA looks good, it's not damaged, it's copied itself well, right? So it checks before it replicates that the DNA is good, and then afterwards it checks it also, makes sure it was replicated properly. Cells shouldn't divide if they have damage to DNA. You don't want that damaged DNA, right, to go from one cell divided, now you've got damaged DNA in 2 cells, and then those would divide, you'd have damaged DNA in 4 cells, right? You don't want that to happen. So one way that cells can check for this is there's an enzyme called P53. When P53 is working as it should, normal P53, it checks the cell, okay, after replication. If there's DNA damage here, right, which this cell has, it says DNA damage, cell abnormalities, right? P53 checks it and it says either stop what you're doing and fix that DNA before you can restart your cycle. So that's option 1. Option 2, you are way too damaged, go kill yourself. That's what apoptosis is. That is programmed cell death. It says this cell has got to die. We cannot have this cell continuing, and its abnormalities are too extreme. We can't be repaired. It's better just to get rid of this cell. So those are what normal P53 will do. If there is a mutation in P53, so the P53 itself isn't functioning as it should. This is an enzyme, right? If this enzyme is damaged, it gets denatured, right? Something happens to this enzyme. It is not going to properly check and control the cell cycle, right? So it either doesn't notice the damage or it's like, whatever, I don't care, it's my day off, right? It's going to let the cell cycle continue. This is how some cells can become cancerous. They're not stopped when there's damage, right? So if you can imagine, if you have a cell that replicates when it's damaged and it just keeps replicating, this is how you end up with a tumor. Cells that have mutated DNA that are going through this cell cycle unchecked, growing out of control. So some cancer research actually focuses on P53. Some cancer research focuses on looking at the DNA itself, right? Some cancer research focuses on the nutrients that cancer cells are using. So there's lots of different cancer research looking at lots of different aspects of cancer, but looking at P53 is one aspect of that. Okay, so that finishes our discussion of mitosis. And remember, when we're talking about mitosis, we are talking about diploid cells. You go from 2 copies of every chromosome, you replicate, you divide, and you end with 2 copies of every chromosome. Meiosis is different. Meiosis is the replication, or really the division, of gametes, sex cells. So this is eggs and sperm. Think about how much DNA you want in eggs and sperm. You have one egg, right? And it gets combined with one sperm. If each of these were diploid, the baby that you got out of that would have 4 copies of every chromosome. That's no good, right? So instead, so that's bad. Let's erase that. Instead, for meiosis, we're going to go from an egg that has only 1 copy of every chromosome with a sperm that has only 1 copy of every chromosome. So the resulting offspring has 2 copies of every chromosome, just like we have in all of our other cells. Okay, so how do we go from most of our cells having 2 copies of it to ending up with eggs and sperm that have only 1 copy? That's meiosis. That's what we're going to discuss here. And meiosis is a little more complicated than mitosis, but you have to understand the pieces of mitosis first. It's got a lot of the same terminology. So meiosis is creating, as we said, haploid cells. That means it has half as much DNA as all the other cells. So if all the other cells have 2 copies of every chromosome, The haploid cells have one copy of every chromosome. All of these are gametes. So in females, the gametes are eggs, and males, the gametes are sperm. Once that one egg and one sperm come together and fertilization take place, you now have a diploid cell again, which will replicate through mitosis millions of times to form an organism, assuming it's a multicellular organism. If it's a single-celled organism, you have one egg, one sperm, and now you have your one little cell. Okay, so our haploid cells include genes from both chromosomes in a pair. So they're unique, right? The eggs don't have only the genes from one of your mom's parents, and your dad's sperm didn't have genes only from one of his parents, right? It's not like you look exactly like your grandpa from your dad's side and your grandma from your mom's side, right? It's a total mush of different, all the genes, which is why you and your siblings don't all look identical. The genes get all mixed up during meiosis. So every egg and sperm is unique. They're not the same as each other. This is different from mitosis, where those cells, after they've replicated, should be identical to one another. The genome is identical in mitosis. It is not in meiosis. In meiosis, it is unique. And as I've said, this only happens in gametes, which means it only occurs in the gonads. So in testicles or ovaries, this is where meiosis takes place. All right, so here we show we've got, two adults, 23 chromosomes, pairs, which is 46 total. It goes through meiosis. The egg has only 23 chromosomes total. The sperm has only 23 chromosomes total. When they come together into a fertilized egg, you once again have 46 chromosomes total. And because, as we've said, and here this step, you have half as many chromosomes. This is showing you, this next picture is showing you this just again, right? We started off with these homologous chromosomes. Homologous means, in this case, that homo means the same. So these are the same chromosomes. This is your chromosome 1 from your dad, chromosome 1 from your mom, right? You have one copy of each in your cells. They replicate, right, for cell division. So you end up, these are your sister chromatids that we were drawing like this before. So there's 4 copies. And then in mitosis, they would split and you would end up, here would be one cell, has a copy from your mom and dad, and here would be one cell, has a copy from your mom and dad. What's going to happen in meiosis is we're actually going to go through 2 different divisions, and so you're going to end up with 4 cells out of that, each with just one copy of it in there. Okay, so meiosis, as I said, goes through division twice. So the first one's called meiosis 1, and the second division is called meiosis 2. The names of the divisions and the steps are the same as in mitosis. So we have prophase, metaphase, anaphase, telophase, like P-M-A-T, okay? There is a replication step in the beginning of meiosis 1. So there is interphase here, okay? That didn't show up very well. Let me change that to black. We do have interphase for meiosis 1, not meiosis 2. Okay, so we've got 2 copies of every chromosome. We have replication. That happens in interphase. Our DNA is replicated. Okay, now what happens in prophase? Our chromosomes condense, right? They squish down. But in addition to the nuclear envelope dissolving, all of that, there's a new step here that didn't happen in mitosis, crossing over. This is also known as recombination. I will show you a picture of that, okay? So in crossing over, those chromosomes are going to swap little bits of DNA with each other. This is how they all end up unique. We're gonna go into more detail of that in a minute, okay? So we have our prophase, everything condenses. Then we have metaphase, things are gonna line up in the middle. but they're going to do it slightly differently than before. And then anaphase, again, they're going to get pulled apart, and then telophase. The cells, are going to divide. We now have 2 cells where we had 1 before. Okay, let's look at this recombination, this crossing over. Okay, first I'll show you the picture, then I'll go back and we'll look through the words. Okay, here's crossing over. Let's say this is chromosome 1 again that we're talking about. This is a replicated chromosome 1. This one's from your mom. And this is your replicated chromosome 1 from your dad, right? You have 1 chromosome from your mom, 1 from your dad. They had been 1 strand, they replicated 2, and they're held together by in the middle here. Okay, so we've got these sister chromatids. In normal mitosis, what would happen is, they would each line up along the middle, get pulled to either side, and you'd end up with 1 pink one and 1 blue one in each cell. You'd have 1 from your mom and 1 from your dad. And they would be identical, right? Every cell would get 1 pink, 1 blue, totally identical. The way that meiosis is different is that before they do that division and get pulled apart, they're going to swap little bits of DNA. Okay, so if this is a long piece of DNA with lots of A's, T's, G's, and C's, right? It's just going to swap out a little bit, let's say here, right? And that exact same bit of DNA on the opposite strand, and they switch. So here you can see this little bit where it has a big B and this little bit where it had a little b. It's the same part of DNA. It's the same gene. A long strand of DNA is just lots of genes, okay? And it just swaps them. So if you think of a strand of DNA as your street, and all of the genes are like different houses on the street. And it's just switching out the last house on the street from one street. It might have a different design than on the other street. It's still the house at the end of the street, right? They just swapped them. So now we've got 4 unique pieces of DNA. This one looks just like it started, right? It has a big A and a little b. This one now is mostly how it started, but it swapped out that last little bit. And instead of having a little b, it has a big B. So it's different than its sister, right? These 2 are sisters. This one swapped with the one next to it. So it has a little b now instead of a big one, and this one looks like it started. So now all of these are unique. And every time things cross over, different parts might cross, right? Maybe the top crosses a different time, right? Or something in the middle gets switched out. So they just switch out different parts. That's crossing over. You end up here with 4 unique chromosomes. Each one's different than it started or than the one next to it. Okay, so this again explains that. Every chromosome has the same genes, but they could have different alleles. An allele is a version of a gene, right? So you can have an allele for hairy knuckles on your finger, or you could have an allele for no hair on your knuckles, right? And so one of your parents might have one of your parents might have another, and they could swish up. Other like alleles that are easy to understand are like a widow's peak, which is whether like at your forehead where the hair comes in, there's a peak in the middle, right? A little point, or if it's more rounded, right? That can swap. So one piece of DNA might have the allele for a rounded forehead, one might have a DNA for a widow's peak of hair, and they could switch places. Okay, so that's how you and your siblings can look different because you get different combinations of DNA. Okay, during interphase, there's interphase in meiosis 1. The chromosomes replicate, so there's 92 chromatids, each one attached to each other. So it looks like 46, chromosomes like this, right? During prophase, in meiosis, those homologous chromosomes, those 4 chromatids, lie on top of each other and swap little bits of DNA, okay? So this results now in having little bits of mom and dad DNA squished together on the same chromosome. So whereas in most of your cells, you have a copy of your mom's and a copy of your dad's, in your gametes, they get squished together into this same chromosome, right? So now there are little bits. If this is your dad's in blue and your mom's was in red, there's little bits of blue on the red one and we can have little bits of red on the blue one. Okay, we've done a little swap. Those parts where they're switched are called chiasma, right? It's like making a like a chimera as a mix-up thing, right? So now the 4 chromatids that we have here, right? All these 4 strands of DNA, they still have the same genes on them. It's still a street with, 20 houses, but the combination of houses might be different. Maybe one street had been all like bungalows and the next street was all like Cape Cod style. And now we've switched it up a little bit. Okay. So we've mixed up the combinations on each strand. And this is the picture we looked at. So your cell, when it goes through meiosis 1, I've just gone backwards a few slides. Okay, you've seen this slide already. It had prophase, metaphase, anaphase, telophase. After this first division, that cell is still diploid. Why is that? How is it still diploid? Well, it still has 2 copies of every chromosome, right? We started with 4 copies. We divided once, we still have 2 copies, just like in regular cell division, right? You replicate your DNA, everything divides, you end up with 2 copies in the end. We're still diploid. We need to go through another division. So I'm going to draw the first meiosis for you once because it's the way that it's slightly different. Let's get this whiteboard, okay? So we have our prophase. This is meiosis 1. This is when crossing over takes place. It's sometimes also called recombination. They mean the same thing. It's recombining the, which genes, which copies of things you have on which chromosome. Metaphase. Everything lines up in the middle. But this is also different than in mitosis, okay? If you think of, remember the cell in mitosis, they all lined up in a nice line like this, right? And then they'd get pulled to opposite sides. That's not what we're doing here, okay? In meiosis, instead, if this is chromosome 1, I'm having like red be your mom's chromosomes and B be your dad's. And there's a little crossing over, right? So things may, okay, they line up like this. So chromosome 1, chromosome 2, chromosome 3 and 4, okay? They line up this way in pairs, 2 by 2. And it's random assortment, which means that sometimes the red is on the left and sometimes the red is on the right. It varies whether your moms are going to one side or another. And for each chromosome, for chromosome 1, in this case, look, chromosome 1, your mom's was on the left. Chromosome 2, your dad's was on the left. Chromosome 3, here again, I put your mom's on the left. Chromosome 4, the mom's on the left, right? It's totally random. But how is this different than in mitosis? In mitosis, every single one gets split. You get one of mom's on one side, one of dad's on one side. They're all lined up, split evenly. Okay, this one, different. When we split this down the middle, these 2 new cells that result are going to have different combinations of genes now. This one has mostly moms on the left, right? And the cell that we're ending with on the right has more of the dad's chromosomes. And yes, there's a little bit of crossing over. Okay, so there's some mix and match going on here. But in general, they're different, right? They didn't divide completely evenly. They didn't split each of these down the middle, right, of the chromatid like that. One side got one set of chromosomes, some mom, some dad. The other side got a different set of moms and dads. Here's how our metaphase is different in mitosis and meiosis. Okay, then we go through anaphase. These got pulled apart, okay, and telophase, we split them. Okay, so now this is after telophase right here that we're looking at. And we still have 2 copies of every chromosome. 1, 2, 1, 2, 1, 2, 1, 2. It's just that they're still attached in sister chromatids, whereas in mitosis, since we had our chromatids line up like this and we split the chromatids, but you had them, the moms and dads were all still here. It's just that they were nicely lined up and each side got exactly half mom, half dad, okay, in these single chromatids. Here, they're still attached. In meiosis, they're still attached as sister chromatids, okay? So we are still diploid after telophase. Still diploid. You still have 2 copies of every chromosome. It's just that they're still attached to each other. They're not single off on their own. So we need to do another cell division. Okay, that's meiosis 2. We're going to divide these again. What we're not going to do is we're not going to go through interphase. We are not going to replicate our DNA because then we'd end up with 4 copies again. We don't want that. Okay, we've got 2 copies and we're going to divide a second time. That is meiosis 2. So meiosis 2, let's go to our pictures here. It always makes me start at the beginning. Apologies there. Okay. Meiosis 2. We do not go through interphase. No interphase. We go straight from telophase of meiosis 1 right into prophase of meiosis 2. Our chromosomes are going to condense again. No crossing over. We did that already. We're done with that. Okay, now this is going to look a lot more like mitosis. We've got all these sister chromatids still connected to each other, left over from meiosis 1. Now they're going to line up nice and neat in the middle for metaphase. Anaphase is going to pull them apart. Telophase and cytokinesis is going to split them. And now we're going to end up with 2 cells. Each one has just 1 copy of each chromosome. So now we have only 23 copies when this is all done. So let's look through this again. Meiosis 1, prophase 1. This is when we've got crossing over. So you can see the reds and blues are kind of lining up and, you know, exchanging little bits of DNA. Metaphase 1, they line up in the middle, but they didn't all line up, you know, all along the line, red, blue, red, blue, red, blue, right along this line, right? They've lined up as pairs with, so if this is, you know, red is on top here and blue's on bottom, red on top, blue on bottom right there. And then here, this one randomly, blue's on top, red's on bottom. Now when they get split, instead of taking just one of our chromatids, one little strand of DNA, you can see they're all still connected, right? There's still these 2 pieces connected together. Half of them get pulled to the top, half of them get pulled to the bottom in anaphase here, okay? And then telophase, we've divided them up. We still have diploid cells, still diploid. You know it's diploid because it looks like this, right? There's 2 copies of each chromosome. If this is chromosome 1, we've got 2 copies of it. Okay, and then we go into meiosis 2. Again, prophase, they've condensed, our spindles are forming, here's our spindle fibers. Metaphase, they're lined up in the middle. This looks a lot more like mitosis, right? Everybody's just lined up in one nice line, spindles attached, pull them apart, and in anaphase, they're pulled apart, and in telophase and cytokinesis, they start to condense again. So we ended meiosis 1, I'm going backwards, with 2 cells here, right? We've got 2 cells. So then when we look at our picture of meiosis 2, it's showing you these 2 cells. At the end, they've divided, and we now have 4 cells. Okay, so for mitosis results in 2 diploid cells, meiosis results in 4 haploid cells. So here's our outcome of meiosis. After meiosis 1, we have 2 cells here. These are about to pinch off from each other. And then meiosis 2, we end up with 4 haploid cells. Each one is completely unique from one another. So when we're talking about male and female in a purely biological sense, I'm not talking about like gender issues here. The female is defined as the one with the larger gamete throughout the kingdom, all life kingdoms, right? So plants have females, can have female and males. Some plants maybe both, but they have female gametes and male gametes, and the female's the bigger one, okay? This is how we define female and male throughout all kingdoms. In animals, the female gametes have more cytoplasm. That's not necessarily true in other organisms, but in animals, the female has more cytoplasm, right? So if we think of an egg versus a sperm, an egg can be very large. A sperm is tiny. It just is a little head of DNA, okay? For eggs only, during telophase, when they're splitting up that cytoplasm, all of the cytoplasm, or the vast majority, is going to go just to one side. All of the mitochondria, the endoplasmic reticulum, all that good stuff that's in the cytoplasm, it's just going to go to one side, and the other one is going to be this tiny little thing that gets like nothing. That's called a polar body. That's just going to degrade. It doesn't actually become an egg, okay? And then both of these will divide again. Again, when this big cell divides, most of the cytoplasm is all going to go to one side. So you end up with 3 polar bodies and 1 egg. So eggs are much rarer. For every meiosis, you really only get 1 usable egg and 3 polar bodies. For sperm, for every churn through meiosis, you get 4 sperm. But that egg, that cytoplasm, is going to also carry that mitochondria and everything else that you need in a cell. Most of that's in the egg. So if we look here at this division, this is just for eggs, right? You start with 1 cell. This is the germ cell. This is the original cell that starts off. It goes through meiosis 1, which is, and you end up with 2 eggs here, or 2 cells here at the bottom, and then it divides again, and most of that cytoplasm is just going to one side. And this doesn't show it very well because it really should show this one as little to start and this one is much bigger, because otherwise there'd be more cytoplasm in these divisions on the right. So you end up, this bottom picture is what you need to focus on. You end up with 1 egg that's haploid and then 3 haploid polar bodies, and these are just going to disintegrate. And just one more slide before we take a break here. I know this is a lot. Meiosis is a lot to cover and you know, contemplate. Why do organisms have sex, right? It works for all other cells just to divide, right? And for prokaryotes, they just have their binary fission, right? They don't need to combine with another prokaryote and then divide, right? They don't need to have sexual reproduction. They just divide. What's the point, right? There must be some advantages to sexual reproduction because there are definitely disadvantages. So our advantages for the offspring are that you're getting alleles from 2 parents, right? So you're getting some diversity. This should say during gamete production, right? There's obviously this crossing over. They're unique, so siblings are different from one another. So there are these advantages that it creates this diversity. This is actually very beneficial because this different combinations of alleles actually help them fight things like for their immune system. So it actually can help fight like bacteria, viruses, things that evolve much more rapidly than the eukaryote itself. So if the eukaryote you think of as this, you know, has this slower, longer life cycle, and things like bacteria or viruses have a quick life cycle, they're constantly evolving. Let's think of this Delta variant of COVID, right? Whereas the larger eukaryote is much slower to evolve. Okay, well, if I've got this new variant coming at me, hopefully my offspring have some new defenses that I don't. So those are some real advantages to sexual reproduction, is that they can mix up the genes and the offspring maybe have some defenses that the parents don't. Okay, so there's our big advantage to sexual reproduction, but there are definite disadvantages, right? Finding a mate, first of all, can be dangerous for animals, right? There could be cannibalism. If we think of like black widow spiders, right? As you're out looking for a mate, you could just get eaten by a predator. There's STDs, right? There's diseases you can get from mating. Also, if you love your genes and your genes are awesome and you want to pass them on, why would you only pass on half of your genes, right? That's a negative. Wouldn't it be great if my kids had all my genes, right? That is the definition of success biologically, is passing on your genes. So it'd be better to pass on more. So obviously, organisms, there must be a real benefit to sex if the disadvantage is only passing on half. Also, that time and energy you spent finding your partner could have been spent doing something else, saving that energy, lazing about, eating, right? Anything else. Finding that partner took energies. There better be some benefits to it. And meiosis can definitely lead to mistakes. That was complicated to explain, complicated to understand. It's more complicated for the cells to go through. We're going to talk about what happens when there are some mistakes in there, okay? So there are some definite costs to sexual reproduction, so there have to be benefits that can balance and outweigh those. All right, so we will stop here. That was a lot of information, and we will finish up the rest of this chapter in the next video.